Good afternoon, distinguished participants and attendees. I, Somiranjan Ghosh, the lead PI of the RCMI funded project from Howard University, sending my greetings to you all on behalf of my whole team. I'll be giving a quick overview on some of our recent findings from, from the RCMI funded project during this presentation entitled Key Biological Pathways of Diabetes in African Americans. Besides my presentation, I shall look forward more towards your prudent advices and comments. Please feel free to have them. Our study participants were recruited from Howard University Hospital with their prior consent and IRB approval. So far as the epidemiological studies conducted and data analysis done, we saw a very strong association of cigarette smoking, obesity, hypertension, which are the key issues that are undoubtedly associated with the diabetes. Where the hypertension was strongly associated with the diabetes, cigarette smoking was strongly associated with the dose-dependent manner, and obesity also associated with the diabetes. Let us see what the gene expression study says. Here, I give you an exclusive example of what knowledge we acquired from the diabetic African Americans. Basically, we started with 54,000 validated gene transcripts that mostly do the key biological functions in humans. After a differential gene expression, we came down to 500 to 700 gene transcripts, which were significantly expressed with plus minus twofold change. Then we ran with these genes, the IPA, to have a knowledge on the top disease and disorder and the canonical pathways, with the top molecules identified. And from our earlier validated studies, we ran 24 transcript gene validation, of which 18 gene transcript passed the validation test, among which 8 genes become statistically significant. The top canonical pathways, that is marked in the red, that belongs to insulin signaling pathways, enzymatic metabolism, cancer pathways, cholesterol biosynthesis, and so on, and which is highlighted in the disease connection in the very right, and with the gene symbols like all these 8 ANSR, ARNT, RRA, TP53, ApoC1, and interleukin 6, GSK3 beta, and E1 are also corroborated as defined by the other studies. In addition, some of this disease, the genes like ApoC1, GSK3 beta, NAE mostly belongs to the neurobehavioral disorders, which is becoming also important under this current study. And what we have a closely looking into them. Here we show the top networks, how the genes are connected with the focus molecules, those are in blue, and the canonical pathways, that is which already portrayed in the earlier slides, like interleukin 6, cell cycle, beta cell receptor signaling, genomatic metabolism signaling pathways, insulin receptor signaling, molecular mechanism of cancer. And in the molecular mechanism of cancer, what we see, the human liver hyperplasia, like in the hepatocellular carcinoma and the cancer pancreatic, cancers of pancreas is becoming more and more predominant, and that is related to the diabetes along with the others. Let's look how these 8 focused molecules behave when we ran a linear regression plot analysis between the relative gene expression of the genes and the HbA1c level of the individuals. As you see, there is a downregulation trend everywhere, means the gene functions are inhibited as the diabetic condition worsens. Take an example of TP53 here, INSR, or the interleukin 6, where they are some upregulated in the individuals, but there's overall trend is downward, means inhibited when the diabetic condition deteriorated. All these observations makes us or give us a very solid foundation on the diabetes and its effects on gene regulation in this African American population. Now this is a matter of time and see how these gene-based pathway molecular pathways holds true in the large-scale population validation studies, which is underway in our lab. Last, not the least, I would say it is a teamwork, and I feel privileged to have a great collaborators in my team. I equally convey my sincere thanks to all of my collaborators and the lab members for their unconditional support to make this work successful, and many thanks to NIMHT for funding this work. With this, I stop here. Wish you a great evening and have a great weekend and some blessed days ahead. Thank you.